Hello again. This is Miss Dagenford. We're going to be continuing our discussion on genetics and meiosis. We're going to look at the stages of meiosis. And so, just a reminder of our terms. Remember, a diploid organism or diploid cell has two sets of chromosomes. A haploid set means, well, a haploid means you only have one set of chromosomes. So what we're going to look at in the process of meiosis is we want to go from diploid to haploid. And that's when organisms reproduce. So meiosis is really a function of reproduction. So in meiosis, we're going to make gametes. And these gametes are going to be reproductive cells. Two examples would be egg cells and sperm cells. They only have one set. So notice we go from 46 chromosomes down to 23. And then when the sperm cells fertilize the egg cells, then we go back up to 46. And at that point, the fertilized egg, the zygote, can divide to become a baby. And that would be going back to mitosis because the cells are just dividing. So what we want to focus on in this video would be the process of meiosis, reducing those chromosomes in half. Now, if we have two sets of chromosomes, that means we have two chromosome 1s, two chromosome 2s, two chromosome 3s, and it's going to be from mom and dad. So we've got one chromosome 1 from mom, one chromosome 1 from dad, one chromosome 2 from mom, one chromosome 2 from dad. And so when it comes time to create the egg or sperm cell, you're going to just separate and give one of each to the gamete. So the gametes are going to get one chromosome 1, one chromosome 2, one chromosome 3, one chromosome 4, and that's going to be distinct from meiosis. So from mitosis, sorry. So meiosis, we want only one set of chromosomes. So let's take a look at how this occurs. Now, before we do that, this is a good time to refresh our vocabulary that we've learned about already. So all of these questions are in slides that we have already gone over. So go back over your slides and type in your answers. And you guys can pause this video and answer them together as a class. So with our meiosis, let's start by knowing that we need to reduce the chromosome number. But as we learned about with the karyotypes, it's really important that the cells need one of each chromosome. Not 0, not 2, but 1. If you have an extra set, that's going to cause problems. And if you're missing one, that's going to be a problem. So we need a complete set of chromosomes. 1 through 23. So how does that occur? Well, first, let me show you a couple of things to notice. One thing to notice is there are some similarities. We go through stages that are somewhat familiar to you: prophase, metaphase, anaphase, metaphase, and telophase. That can be really frustrating for students, and we're gonna go over very, very clearly each stage. The other thing to notice is in meiosis 1, these chromosomes are clinging to each other. Same thing in metaphase 1. Suddenly we have chromosomes pairing up together, and that's going to be a hallmark of meiosis. The homologous pairs are going to line up in order to then be distributed to each daughter cell. And you'll see it in the diagrams. So notice we have two stages of meiosis, meiosis 1 and meiosis 2. Now in this picture, the cell has 6 chromosomes total. 3 blue and 3 red. Now notice in this diagram, again, they're kind of clinging to each other. One blue, one red. One blue, one red. One blue, one red. That's going to go back to the idea that you've got one chromosome 1 from mom, one chromosome 1 from dad, one chromosome 2 from mom, one chromosome 2 from dad, and then 3 from mom, 3 from dad. And so what ends up happening is we want to get one of each type to the gametes. And so you're going to see them lining up and then being distributed. But the key to notice in meiosis is that it's the pairs that line up and then the pairs that separate in this first stage. That's going to immediately reduce the number of chromosomes in half. So let's take a look at each one. So in prophase 1, there are going to be some similarities to what you've learned before. In prophase 1, the nuclear envelope breaks down, the chromosomes condense and become visible, the centrioles have duplicated and they're migrating to the poles. You can see those right here. And then the centrioles start building the spindle fibers. You can see them in the yellow lines. The key differences, and I bolded it and capitalized it here, homologous chromosomes pair up. So here they are kind of clinging to each other. And in fact, so much so that they can actually swap pieces of chromosomes. And that could really kind of change how things are inherited. But notice the chromosomes are paired with its homologue. And that's going to be very different from mitosis. Remember, we're doing meiosis, which is the reproductive cell division. So in prophase 1, the homologous chromosomes pair up, and they're going to start moving to the equator. So in metaphase 1, the nuclear envelope is gone, the centrioles are at the poles, but notice here's the big deal. The homologous chromosomes are at the equator. The pairs are at the equator. In all of these diagrams, the pairs have lined up together. And this means that when they separate, you may have gotten the chromosome that has blue eyes from your mom, but your sister may have gotten your mom's green eyes, and therefore your sister has green eyes and you have blue eyes. Or maybe your mom has a brown eye gene and she got the brown eye gene and you got the blue eye gene. And so that's why you have variation within your family members, because of this difference in creating the gametes. Now, the homologous pairs will sort randomly along the equator. So for instance, you could have blue eyes or brown eyes, or you could have straight hair or curly hair. What if they sorted so that the blue eye is on the left, but so is the curly hair? That would mean that the one egg cell contains blue eyes and curly hair, and the other egg cell would have brown eyes and straight hair. And so, again, this is why your brothers and sisters might have slightly different looks, because one of you may have gotten your mom's green eyes, your brother may have gotten your mom's brown eye gene. This is called random assortment. And notice how you have two possibilities here. You have the blue chromosomes lined up on the left and the red lined up on the right. Over here, possibility 2, you have a blue and a red, and then a red and a blue. That means that the gametes that you make are going to be ever so slightly different, and then that gives rise to slightly different offspring. If you've ever noticed the different puppies and kittens in a litter, this explains them. They get different traits from mom and dad. Here's an example where we have 6 chromosomes, 3 homologous pairs, and depending on how they sort, you could get different traits being given to the puppies or kittens. Now, in anaphase 1, the nuclear envelope is gone, the centrioles are at the poles, the spindle fibers are moving the homologous chromosomes to the equator, and then the 2 daughter cells get um, uh, only half of the chromosomes. Here's where the reduction of chromosomes occur. So in telophase 1, the nuclear envelope gets remade. Cytokinesis occurs. 2 cells are made, but both are haploid. Notice now we've got 1, 2, 3 chromosome on one, and then 1, 2, 3 chromosome on the other. Now, in prophase 2, sorry about that, in prophase 2, the nuclear envelope is breaking down. Again, the centrioles are moving to poles again. This time, the spindle fibers are going to connect to individual chromosomes. And again, notice we have split them in half, meaning we've only got 3 chromosomes in each cell. And we are going to set ourselves up for metaphase 2. In metaphase 2, now the chromosomes themselves line up, not the pairs, the chromosomes. And so what we're ready to do is split the chromosomes in half. And but again, notice we've got 1, 2, 3 chromosomes. So that's metaphase 2. In anaphase 2, the chromosomes are now split in half. So the chromatids are getting pulled apart, and now each chromatid is called a chromosome. And so now we have 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. But they're going to be split into 2 daughter cells, and that's where we're still going to get half the chromosomes in each of the daughter cells. So in telophase 2, remember there's still only 1 set of chromosomes. The nuclear envelope is rebuilt. The centrioles are at the poles. Cytokinesis occurs. What we end up getting is 4 haploid cells that each have 3 chromosomes. Remember, we started with 6. So that's where we get our 4 gametes from. Now, we've got some helpful videos that you're going to watch, and then you're going to have a chance to practice matching each stage for each set of meiosis. And these are the pictures that we started with in a previous slide. So you can, there we go. So you can match them up that way if you want to go back and look. So that should help you walk your way through meiosis. Remember, the big deal is reducing the number of chromosomes in half.